Um, the talk I'm going to give is based on the paper that's coming around right here. I was asked to give this, to write this paper by Howard Broman, who's the editor of the ICES Journal. And then people at the university got news of the paper because I mentioned the Canary Islands in there. I talk about it's a saga from my life coming from the Thoreau Woods in Massachusetts to the Canary Islands. So the university picked up on it and asked me to write a short summary. And then Pili found that and asked me to give this talk. So that's the genesis of this particular talk. Now, notice in the title, you've got Henry David Thoreau, you've got Trump, and you've got scientists of us. That's us. What's the relationship here? I'm going to try to explain who Henry David Thoreau is. I'm going to just briefly say something about Trump, and then give you some examples on how you can use the philosophy of Henry David Thoreau to pursue your science. Okay, now down below here, this is a cartoon of Henry David Thoreau right here. He was a naturalist in Massachusetts 150 years ago, and he did an incredible amount of work with no money and in 20 years, because he died at about 46. All right, so let's go to the core of this talk is that we're under siege now as scientists because of Trump and the philosophy that surrounds Trump. This right-wing, conservative, fascist kind of thinking that is plaguing Europe and the United States. Trump is the worst of it, and he's really a problem. He is rolling back the rules and regulations that have been set up in the United States to protect our water, to protect our air, to protect the people of the United States. The age of Trump, at least my characterization, is ignorance of history. Ignorance of the role of fact-based decisions, which are the basis of our civilization. Ignorance of the importance of free press, which keeps our democracy alive. Disrespect for universities and research communities. And he's ignorant of climate change. He thinks it's a hoax, think that as scientists, we're just having fun playing this hoax on the population. This guy is really bad news. He's negated for the United States the Paris Climate Agreement. Luckily, the rest of the world is fighting strongly to maintain the agreement that was made in Paris and trying to keep climate change at a minimum. But what Trump is doing in my country is that he's allowing the industries to almost repollute the rivers that have been cleaned up in the last 30, 40 years. He's allowing the coal plants to start polluting the air again. And this is a terrible, terrible thing for our people at a social level, but for scientists it's terrible also because he's cutting our funding and undermining the people's confidence in science. Henry David Thoreau is sort of the opposite of Trump. Here was this guy, a free spirit, citizen scientist from 150 years ago. He's Harvard educated, he had a good foundation, but he was a real maverick. He decided to build his own house, this little house here, it's a replica of it, in the woods in Massachusetts near this pond called Walden. The reason I tuned in with this guy is I grew up in these woods and spent my really formative years living a life like Thoreau's. That's another story which I could tell you, but actually we lived a life like Thoreau's, incredible. But what Thoreau did, the point is, that's what's important, is Thoreau wrote some really, how do you say, impactful papers and books. This book right here, Walden, is a book about his philosophy of, basically it's an anarchist kind of philosophy, but it is really important for American thinking. And then he wrote this essay, really, Civil Disobedience. And he wrote it when he was in jail. And he was in jail because he wouldn't pay his taxes. He disagreed with the war effort. The Americans had a war with Mexico in 1848. It was an unjust war. It was an imperialistic kind of war. Nothing that I can be proud of, that's for sure. And he wasn't proud of it either. And he says, I'm not going to pay my taxes. So his friend, who was the jailer in the town, put him in jail. And so while he was in jail, he wrote this book, a small essay, Civil Disobedience. But that little essay provided Gandhi the philosophical foundation to declare independence of England, from England, and actually win the independence of India from England. And Tolstoy, based on this civil disobedience, was able to free the serfs in Russia. And Martin Luther King, using the same ideas, the same essay, was able to be successful in the civil rights movement in the United States in the 50s and 60s. And it really gave all of the African-American people in my country another lease on life. They thought they had it 100 years before at the end of our Civil War, but they didn't get it, really. But it was through Martin Luther King, based on the civil disobedience idea of pacific, non-violent resistance against the tyranny of government. Oh, that's the kind of stuff this guy did. He did it in 20 years with no funding. That's the point. No funding at all. Okay, this is now just a summary of what I just said. Basically, he wrote Civil Disobedience, he wrote Walden, but he practiced it. He practiced self-reliance, political activism, and citizen science. He laid the foundations of limnology, botany, geology, ecology. There were other people, a lot of other people in the world, but he, without any funding, made basic measurements in these fields. And Nature Conservancy, he was one of the founders of it, and his observations in the woods, observations of when the plants flower, when the leaves fall, when the snow comes, when the ice breaks up on the lakes. He wrote this down daily in a journal. And that has provided, as we'll see in a minute, the baseline for climate change studies in Massachusetts. And a fellow, this guy Primack at Boston University, not only did he write this book, but he's got a number of papers based on this data from Henry David Thoreau of 150 years ago. So here's a guy, no money, no official encouragement, making observations daily in the woods, which provided a baseline for climate change. It's an example for all of us. This is some data from Primack. It shows Thoreau's early observations of the first flowering of the plants in the woods around Walden Pond. And this is, he basically shows here that you would get the flowering around the 15th of May. And Primack and his colleagues, Miller Rushing and other colleagues, showed that actually now you advance this down to about 5 of May. So basically you've gained a week. Okay. But without that kind of baseline data, they wouldn't know that. They wouldn't really be able to trace the climate change all the way back 150 years. Now, the lessons from Thoreau, and these are lessons that I think, and especially for you students coming along, these are things that are really important. Follow your interest and curiosity, regardless of whether you're funded or not funded, regardless of whether people encourage it or don't encourage it. If you believe in it and you want to do it, follow it. That's what Thoreau did. He did everything based on his own internal curiosity. Don't give up if you're not funded. That's for modern. In those days, they didn't have any funding, but we do. And many times it's discouraging. Madrid doesn't want to give you the money. Washington doesn't want to give you the money. Many times you can't get the money and you feel like giving up. Don't give up. Do what Thoreau did. Make your observations every day, and pretty soon, eventually, you'll succeed. Don't give up if your paper is rejected. In our group, we just had a rejection today, and it's discouraging. And it's really, you know, it pushes you down. But don't give up. Rewrite the paper, add some data, change something, find a foil, and do it. You can do it. Write every day. Now, I wish I did. I don't. Thoreau did. Every day he wrote in his journal. And his journals, now they're in one of the libraries in Boston. I don't know whether it's Harvard has it or not. But they're still mining his journals for new information. Believe in yourself. You know, you'll find a lot of people that say you're stupid, you're dumb, you're misinformed, and all kinds of nasty stuff. I mean, this happens in the scientific world. Don't believe them. Just stick to your guns, follow your interests, as I said up in the top. Follow your interests and curiosity, and believe in yourself. Okay? And look, what happened here? This is another thing. Be honest and let history be the judge. When you make observations, when you do experiments, many times you'll get a result which doesn't jive, doesn't agree with something from the literature, doesn't agree with something your colleagues say. But don't change it. You know, you made that observation. The best thing to do is go back and check it, do it again. And if it comes out to be the same, if it's different from other people's, stand by it. Many times, this kind of observation, which disagrees with past observations, opens up a whole new world in science. And there was I've lost his name. But anyway, there was a philosopher that will come to me, that basically pointed out it's the disagreements in science, the disagreements in your observations, which leads to the advances in science. Okay, now, here we are in Las Palmas, the University of Las Palmas, Gran Canaria, and what does Thoreau have to do with us? All right. In our group, my Gomez's research group, the EOMAR group, in the part that we have a number of different projects, but in the areas that I'm working in, we've made a number of advances, a number of new concepts in the last couple of years, and this isn't just me, it's due to the efforts, the teamwork of all of us, but we've laid down some new concepts which were had zero funding. They're based on playing with the data, thinking about it, reading, and coming up with new ideas and new concepts. And I'm going to list them right here, because these concepts are not in the textbooks. All right? They're new concepts that have been developed right here. One is modeling respiration. Now, in biology, we don't have mathematical equations for the processes, photosynthesis, nitrogen fixation, respiration, ammonium excretion. We don't have equations the way you have equations in chemistry and physics. And one of the things that we're interested in doing is actually finding out what are the fundamental equations that lie behind these biological processes. And we found a way, using enzyme kinetics, to model respiration based on first principles. I'll go in and show you that later. The second thing is we found a way to calculate the ATP production, the heterotrophic energy production from the electron transport activity and the relationship in the electron transport activity between oxidative phosphorylation and oxygen consumption. We've developed a concept called the nutrient retention efficiency. It is the ratio, here I've got it right here, it's the ratio of the respiration in a layer of the ocean divided by the carbon flux that goes into it from the top. So the nutrient retention efficiency is that ratio, and what it tells you is the potential for regenerated productivity in a body of water, especially if it's close to the surface. It can tell you whether that water can support productivity, not new productivity, but regenerated productivity in the weeks to come. So it's an index of future productivity. The curvature, okay, what is a curvature? Okay. In a respiration equation in the ocean, if you have respiration as a function of depth in the ocean, here's an example right here. Here's depth down here, here's the respiration equation plotted out. This has a curve. If this B here was zero, wasn't there, got to zero, this would be straight up and down. That B governs the curvature. But we found out that B also controls and governs the flux of carbon through the ocean water column. It controls the nutrient retention efficiency. These things, this is a P. This is the nutrient retention efficiency. As the B, this number here gets high, the nutrient retention efficiency gets high. As the transfer efficiency of carbon through the water column gets low transfer efficiency, this B gets high. So that curvature controls carbon flux in the ocean. Okay? That's a totally new concept. Carbon flux from respiration. That's not really a new concept. But what's new about our work is that we've developed a way to calculate the equation so that you can get carbon flux at any point in the ocean water column from by knowing the respiration in the water column. You can calculate the carbon flux from that. And I think the last one is this is cool. You wouldn't suspect this, but the burial of carbon in the muds of the bottom of the ocean and the respiration of those muds, 5,000 meters down, is dictated by the curve of respiration in the water column above it. And if you know that curve, you can calculate the benthic burial, carbon burial, and the benthic respiration. There's a trick. It's a mathematical trick, but it works. And I'll show you that as we go along. But those are 6 basically new ideas and new concepts that we've developed here at the University of Las Palmas, Gran Canaria. And they were funded at a very, very, very low Thoreau type level. All right, and the first thing here is modeling respiration. And I'll try to do this fast here. Basically, we found out that if you use an enzyme kinetic kind of model, this is respiration here, this substrate is like the food of the organisms. If you use that kind of equation, you can predict the respiration in a bacteria culture from the time you start the bacteria culture to the time the bacteria are starved. Okay, what happens here is if you take a bacteria culture at time 0 and you give it pyruvate, you could give it glucose, you could give it anything, but we gave it pyruvate. Okay, the bacteria eats up this pyruvate and turns that pyruvate into cell mass. And the cell mass that it produces, this is down here. Let me see, I don't have the next one. This work was worked out a lot by one of the physical oceanography students here in Las Palmas, by but what you get is that, let me see if I get exactly where it was. The pyruvate goes down, the cell mass of the bacteria goes up. It's the mirror image of this of the food source. The ETS, which is the potential of respiration, it's the potential metabolism of the organisms. That sort of follows the biomass more or less, especially in the growth phase. It starts to get a little flaky here, but that's basically a measurement problem of this particular experiment. And then the respiration looks like none of those things. The respiration, the oxygen consumption, the CO2 production of the bacteria culture grows high in the growth phase and then drops down to almost nothing when the cells are starved. And the point of this whole is that you can calculate this respiration at any time on this plot, any time in the life of the culture. If you know this measurement here, which is this one, the green, and if you know the food source, and that's this right here, you can calculate that respiration. This is another experiment using the same equation though. This is your measured respiration. If you use the model that I've said, this enzyme kinetic model, you can actually see the respiration. You can calculate the respiration, which gives you the same form. This is not in the textbooks, all right? This is not understood by 99.9% of the practicing biological oceanographers. We now have a way to calculate respiration, but from enzyme kinetics. Okay, now, the other 5 concepts were worked on by the team that I've got here. These are all our graduate students, and Mike Gomez here. And we put together the other concepts that I mentioned, the heterotrophic energy production, the nutrient retention efficiency. Now, the heterotrophic energy production is the ATP production in the ocean. Ecologists, for the last 50 years, have talked about energy production. Energy here, energy there, transfer of energy through trophic levels. They never measured it. All they ever did was measure biomass. Biomass is not energy, even if you bring in Einstein's equivalence, equals mc squared. It's not the same thing. You know, we need to be able to calculate energy at different trophic levels in an ecological system. All right, we can do it now, but it's no big deal, actually. This guy, Ochoa, Severo Ochoa, Basque fellow who gave up on Franco in the 40s, after the Civil War was won here, Ochoa said, no puedo más. And so he left for New York, And there in New York, he laid down our understanding of the relationship between the production of ATP, the energy of the cell, and the consumption of carbon and the utilization of oxygen. And he found a 3-to-1 ratio, which seemed to hold in all cases between the respiration and the ATP production. Later on, Peter Mitchell explained the coupling, the association at a biochemical level between oxidative phosphorylation and oxygen consumption. Here's Peter Mitchell here. I don't have a picture of Joel, not sure. But both of them ended up by winning Nobel Prizes. We're talking about really smart people. This guy's a Brit and Joel is a Basque. But what they lay down is the relationship between the production of ATP by the ATPase in the membrane of either a bacteria or the mitochondria. It's universal. Not only in respiration, but in photosynthesis. This is one of the most ubiquitous enzymes on the planet, all right? But it's coupled through this mechanism here to oxygen consumption and the electron transport system, because the electron transport system not only pumps electrons up through this membrane, but it pumps protons across the membrane. And this proton gradient across the membrane actually drives this ATPase like a motor. It's actually a small molecular motor. And anyway, the point is, all the ecologists in the last 50 years, and all the marine biologists, all the physiologists, could have been calculating ATP production, but they didn't. It could have been calculated simply from their respiration measurements. All through the literature, you'll find papers after paper after paper of oxygen consumption in copepods, in euphausids, in benthic organisms, but nobody tried to use Ochoa's work to calculate ATP production. Here in IOMA and Mike Gomez's group, we've developed this, and we are now making calculations of ATP production in mysids, in jellyfish, in the water column, in the benthos, and we're laying down. Now it's so new that we just don't know the temporal and spatial distribution. We don't know anything about the ATP production, but it's a totally new calculation, but based on old thinking from the ecologists. Okay, that's that. In the ocean, this is what the heterotrophic energy production looks like. This is a cross-section across the Peru current. This is Pisco, Peru, 15 degrees south. This is out over, let me see, the trench, the Peru trench is in here. You get way out over into a plateau beyond the Peru trench here. This is the heterotrophic energy production contoured in joules per cubic meter per day. That's the same as a millijoules per liter per day. But now we've got basically the energy production in the ocean water column. We didn't have that before. Nutrient retention efficiency, another concept. All right, as I said, it is the ratio of the respiration in a layer in the ocean divided by the flux of carbon going into it. This is the diagram. This is the diagram here. Now, what it looks like in the ocean is this. This is a station over the edge of the Peru shelf, down to 500-600 meters. And you'll see the nutrient retention efficiency is high in the euphotic zone and high down in the nephaloid layer just above the bottom. It goes through a minimum in the mid-water column in the oxygen minimum zone. It's exactly the mirror image of the carbon flux transfer efficiency. You'll hear in some of the classes in biological oceanography around here, you'll hear people talk about carbon flux, carbon flux, carbon flux. There is a measure called the transfer efficiency, which effectively is the difference of carbon fluxing between two layers divided by the carbon flux that flows into the top. And that's what this number here is. But the nutrient retention efficiency is the mirror image of that. The two together equal 100%. So you can calculate one from the other at any time. But the way we calculate the nutrient retention efficiency, we do it from a respiration profile in the water column. That was never done before. Now, the bee. I mentioned earlier the bee here, and I basically told the story before about the importance of the bee. But this is what I call the Dixie diagram. It shows a perfect correlation between the nutrient retention efficiency and the curvature of the respiration profile. The transfer efficiency shows the opposite. It's an inverse relationship. Carbon flux and respiration. For those who don't know, this is sort of a diagram of what carbon flux is in the ocean. You have all these little particles in the ocean. It's called marine snow. And it slowly sinks down through the water column. And as it sinks down through the water column, the bacteria colonize each particle. And they start to eat it away. They start to utilize the carbon, producing CO2. Let me see, they'll be producing CO2, they'll be producing ammonium, they'll be producing phosphate, they'll be producing silicate, and that happens all the way down through the water column. But the carbon flux going through this layer, so minus the carbon flux going through this layer, is the respiration in this layer. Okay? And this is something that a lot of people find a hard time understanding. This is a diagram of it right here. And the respiration between layers in the water column equals the difference in the carbon flux between the top and the bottom of that. And if you integrate all of the respiration from the bottom to the top, you get the carbon flux going through the top. And that's what this equation here tells you right here. You take the respiration profile in the water column, you integrate it from the top down to the sediments, you get this equation here, which tells you what the carbon flux is coming here at the top. And if you change your parameters here, this is top and this is sediment, if you make this, say, to 200 meters, you'll get the carbon flux that flows into the top that gets metabolized in those 200 meters. This is the thing that we've developed here that is not in the, it's actually, except in our work, it's not in the literature and it's not in the textbooks. What does this look like? This is carbon flux in the ocean along the same transect. This is in Peru, 15 degrees south. You see this really strong pulse of carbon flux here. This turns out to be just over the shelf edge, where the biomass, the carbon, the chlorophyll, and the particular protein is at a maximum. The upwelling off the Peru coast has a maximum someplace in here, but that's the upwelling means the water's coming up. The nutrients into the surface waters is at a maximum here. But it takes a few days before the biomass goes to a maximum. And that comes out over here, and then it starts to fall down in this burst here. Benthic burial and benthic respiration. This is the one I love so much, actually, because if you take the respiration curve and you integrate it to infinity, you integrate it to infinity from the bottom of the euphotic zone, so that's about, you get a fairly large number. If you integrate it from the sediment layer to the bottom of the euphotic zone, you get a smaller number. The difference between those two is the carbon burial and the benthic respiration. It's a trick in the mathematics, but it works. Where you see high levels of carbon burial in the bottom and benthic respiration, given by this black number here, you find minimal nutrient retention efficiency in the water column above. Here, these different sectors here represent the epipelagic nutrient retention efficiency. This is the upper mesopelagic, lower mesopelagic. This is the bathypelagic. So these are different layers in the water column above the sediments. And what this shows you is that when the nutrient retention efficiency, in other words, when the respiration is really high and the bacteria and microorganisms in the water column are recycling the carbon that's falling down, not much gets to the bottom. And so you get a low benthic burial, benthic respiration, when the nutrient retention efficiency in the water column above is high. So it goes, it's as you would logically think. But it works out very nicely, and it all comes from this equation right here. All right, so just applying Thoreau's kind of thinking of going ahead and just working, even though you're not funded, we've been able to improve respiration modeling, develop the concept of ATP energy production, developed carbon sections from respiration, developed the concept of nutrient retention efficiency, learned that the exponent in the respiration profile controls carbon flux and nutrient retention efficiency, and developed a way to predict benthic burial and benthic respiration from integrating the respiration in the water column above. Okay, I was asked, PD asked me about my history, about my formation, where I came from, why did I go the way I went. It turns out I went to MIT. This is MIT. Oh, well, this is all MIT here. This is the Charles River over here. This is Boston. This is Cambridge on this side. Harvard University is down, where is it? This is Mass Avenue. You go down the road over here, and someplace over here is Harvard University. So the two places are very close together. And for 100 years, they've been competing, like Oxford and Cambridge in Britain. They were competing with one another. They're still doing it today. But they have a lot of good programs of collaboration between one and another. So the question was, why go to MIT at the university, and to the University of Washington? Because I went to the University of Washington after I went to MIT. Well, as a kid in high school, I loved history. My dad said to me, "Yeah, but you got to write books." And I thought, I'm not a good writer. I knew it. Writing's hard. I do it, but it was hard. So I nixed the idea of going into history, and went to MIT. But I went there partially because of this. MIT had a connection with Woods Hole Oceanographic Institute. And their professors would go back and forth, and students could do theses down at Woods Hole. And so I thought, hey, that's good for me. I was going to do that. As I say here, I was a problem solver, not a writer. What I didn't know, and this is something for you young people coming along, you really need to realize that you got to write. And the lab work is critical. Working on the computer, doing modeling, doing analysis is critical, but you got to write. Thoreau said. At the University of Washington, I went there. I had to admit it, I went there partially for non-scientific reasons. The forests in western Washington are spectacular. Trees grow to 100 meters high. The rainforests there are just gorgeous. In addition, you have these mountain ranges on both sides of Seattle. The Olympics on one side with the Mount Olympus, the volcano on one, and then on the other side you have Mount Rainier. On this whole thing. It's just a gorgeous place. We'll get into that in a minute. But so I went there because I love the place, I love the setting. But in addition, the University of Washington, at the time I went there, had an incredible spectrum of biological, chemical, physical, geological, oceanographic courses. And they were oceanographic courses. And the professors that taught them were not chemists, biologists, physicists, geologists. They were ocean chemists, ocean physicists. They understood the ocean as a system. They had all studied and worked with the writings of people like Sverdrup that laid down the foundations of oceanography. And I was attracted to this. Oceanography to me was an integration of all the sciences. And I like biology, but I really wasn't strong in biology. I actually liked chemistry better. I loved physics, but I wasn't that abstract. I mean, what was neat about oceanography was it integrated all the sciences. Okay, back to MIT. One of the cool things about MIT is it never stops. If you go there on Sunday, you'll find people working in the lab. You go into the libraries, people are in the libraries there. You've got, here's a background. Mine's got a different But the reality is, it's gorgeous. It's August. No vacation. Yeah, you can take your vacation, but you take it when you want to. You don't have to take it in August. And the school doesn't shut down in August like this place does. I mean, I love this place, but it's hurt our students a couple of times because it shut down right in the middle of their experiments. It's hurt. MIT never does that. It's open summer, winter, fall. It's Sunday, Saturday. All the time, it's always going. If you want to study, you want to get a book, there are places on the campus that you can go. Here is under this dome, they call this the Great Dome here. This is looking up at it. Down below it, you have a reading room here in the daytime. At night, it's still functioning. Okay? So it's a really great place if you really want to study, if you really want to do lab work, and just focus on your academic kind of stuff. It's also great because it's connected to Woods Hole. This is Woods Hole here. And Woods Hole is a super place. It's really great. It's got libraries over here. There's the Marine Biological Laboratory associated. There's a lot of different schools that have activities down here. And many universities, professors of many universities in the East spend their summers here. So it's a really, it's a very academic, cosmopolitan, ocean-oriented research center. I did my senior thesis in this building here, and in the laboratory of John Canaria. It was great experience. Friday Harbor, the University of Washington is another place, another world. Here it is. This is the University of Washington campus. This is the main quad right here. This same place here. In the distance, you can see Mount Rainier. It is a 3,600 meter volcano that can still erupt. And up at this end, out of sight over here, is Mount Baker, another volcano. It's not as tall as Mount Rainier, but it's a volcano. And all the way down to California, there are chains of volcanoes. This is a fantastic place. The forest, this is Lake Washington right here. On the other side, or this side over here, would be Puget Sound, which goes down to 600 meters. It's a very deep fjord. And so you've got the ocean, you've got the lakes, you've got the mountains, the forests are incredible with the trees that they're set up to 100 meters high. It's a really gorgeous place. It has Friday Harbor Marine Laboratory a few hours to the north. A great place is the library of Friday Harbor, the dock. The plankton is so rich that you can go down, put a light out, and then go down at night and scoop out plankton and do work on it in the laboratory. So that's part of the university here. The other thing that's really cool about the University of Washington is its oceanography department is right on the wire. This is the teaching building of the oceanography department. The faculty building is right about in here someplace. And then on the other side of the faculty building is a dock with the T.G. Thompson tied up in front of it. And two smaller boats, which are used for Puget Sound work. This one, and on the other side there's another one. And one of the advantages is that as a grad student, if you're planning a cruise, you can daily go onto the ship, set up your laboratory, get everything organized. And then in the basement of the office building, which is right about in here, this may be part of it, yeah, there are ateliers, workshops. You can go in and build equipment, you can fix equipment, and it's all right there, and your office is really close by. So the proximity is fantastic, and it gives it a very great advantage. This place, to my surprise, just was ranked #1 in oceanographic institutes in the world. When I went there, it wasn't that at all, but it was recently ranked there. But now I understand why, because the facilities are fantastic, absolutely great. Okay, we could do that. On this island, we could do it. But, I mean, it takes will and vision to do something. Anyway, the other places I've been to and worked in my life are Bigelow Laboratory in Maine, the National Science Foundation in Washington, University of Aix-Marseille in France. This is Lumini, the Lumini campus, with this beautiful mountain in the background. And the Institut Ries-la-Montagne in Canada, a gorgeous research center on the banks of the St. Lawrence River. The problem is, you've got 7 months of 4 meters of snow. So, I mean, unless you like snowshoeing and ski de fond, it's a tough place. But it's a beautiful place. Okay. This is my trajectory from graduate students to today. This is the publication record, a 3-year running average. You'll notice while I've been here, everything has been going uphill. I mean, and I really credit this place for allowing me and these publications are co-authored with all our team, our researchers, Maya Gomez and Eco and all the other people in our team. But it's parallel in a way to this curve in the beginning of my career. when I had a similar team. It was my responsibility. Now this team is Mike Gomez's team, but I'm part of it. And you see the same kind of parallelism. Both were universities. Both had students who were eager, the students who could write, the students who wanted to do research. That is really key. In the places, this is really a sad example. When I moved to Quebec, I was at this gorgeous research institute here. But the philosophy of this research institute was don't publish. We were told by the director there to write reports for the politicians in Ottawa. Now, even though internally my code was to write, I wanted to do basic science, the ambience all around was so negative that it had a major impact on my productivity. So this is, the ambience around you is really crucial. I had stability in funding. I had a great research team. Now that's another factor. The difference here, the research team here, everybody on the team could write. Not only could they do the science, not only could they read the literature, but they could write. Here, I was the only guy that could write. The other ones, everybody on my team, not only, well, they could write, but they didn't really want to. Why, partially? Because writing in science means writing in English. And the Quebecois didn't want to have anything to do with English. And that made it really, really, really hard because all of the work that we were doing fell on my shoulders to write up. And it was just too much. Okay, but I could go through these elements. What time is it now? Okay, I'll have to go faster. I could go through each of these things. This is why I went to France, back to Bigelow Laboratory. This is moving in Spain. I'm retired, early retired. Actually, my official salary from any source actually ended right here. But that doesn't really mean that the productivity ends, because what's needed is stability. And I have stability here, and I'm able to produce. The students are really critical. The lessons learned over the years, I'll try to do this, at the university, it was just a real lesson to see that real oceanography is integrating all the sciences, everything we do. From my work with the upwelling program, I learned that the co-authoring is great. One of my professors, I won't give his name, said, no, you must publish singly. It's a single author, because then when you go looking for a job, nobody will know whether you did the work, whether you didn't do the work. They won't know how much you're responsible for. But in oceanography, you don't work alone. Somebody does the physics, somebody does the chemistry, somebody does the biology. You have a team, and there are people responsible for all different parts of it. If you try to publish on your own, you end up by exploiting other people. That's bad. Don't ever want to do that. So, and I saw it very clearly, and now we know, the more you co-author, the more citations you get. And if you co-author international people, you get even more citations. So now that everything is based on citations, it behooves us all to co-author. And the reality is, that is the truth of what science is all about. You work together as a team. Everybody does his or her part. Okay, the Bigelow Laboratory, this is sad. I learned the lab director needs independent funding. If the lab director doesn't have independent funding, he or she starts to compete with the people that he or she is directing, and then you get really bad politics, bad human relationships. The director has to be independently funded and stably funded. We didn't have that Bigelow Lab. Today it does. At the University of Aix-Marseille in France, I saw very clearly that top-down science, driven by managers in a different place, in that case it was in Paris, leads to minimal creativity in science. I'll add, well, let's see, I don't want to go further on. Well, just the opposite of that, when I was at, and that was similar, well, no, I won't, Instruments La Montagne was a different story. But at Ciencias del Mar in Barcelona, and this is kudos to Spain, the scientists are, the research programs of the scientists are driven by the scientists. And there's a lot of creativity in Ciencias del Mar in Barcelona, completely different from the laboratory in Marseille. Okay, what I learned at the National Science Foundation, this is some good things and some bad things. I did some studies and saw that if you can fund on a specific project a research team of about 5 people, a PI, a close colleague or postdoc, a couple of graduate students, and maybe 1 technician, if you fund that, you will get the maximal output. In other words, maximal paper research production per person and research production per euro. And I did that experiment. I had in front of me the dossiers, the files of all the US chemical oceanographers. I had them, and I was able to analyze all this data, and that's one of the things I found. I didn't publish it. I should have. But it was really a great demonstration. The other thing here is, this is sad, pianist power. Program directors, and whether they're in Madrid or Washington or Paris or London, or wherever they are, they have incredible power, really subtle power. And the power is they know the research community. They know who your colleagues are, and they know who your enemies are. And when you send a proposal to them, if they like it, they can send it out to review to your friends. If they don't like it, they can send it out to your enemies. And nobody can say anything. There's no defense against that. There's no court. They have the power to do that, and they do. They play the research community the way a pianist plays a piano. They know the positive people over here, the negative people over there, and they can do that. And you have no control over that. Regardless of what country, it's the same. And that's something that needs to be addressed. I don't have the answer, but it needs to be addressed in the future. Canada, really nice demonstration. I took a management course and I learned everybody counts. You've got a research team, you have to make a decision. You get the best decision if you consider the thoughts and the knowledge of everybody in the group. Now that takes time. It's slow and it's hard. But they demonstrated that to us very, very clearly in a lab. We set up a laboratory. We spent a whole afternoon, and we demonstrated it very clearly. The best decisions were made when everybody's opinion and knowledge was considered in the decision. And that is really a democracy kind of thing. But it was demonstrated to me very, very clearly. That's one of the reasons why I believe so much in teamwork and no exploitation. Consider everybody, try to draw the positive out in everybody. What I learned at the Institut Bristol Montaigne, as I said, all teammates need to write something because otherwise it's too much on the leader. At Ciencias del Mar, Barcelona, I learned about multilingualism. And I learned that in 1970, a long time back. It was so clear at an oceanographic conference who was able to take advantage of it. There were French people, German people, American people, English people, Spanish people at that meeting. And in the Spanish community, you had the people from Madrid and Vigo and Barcelona. The guys from Barcelona, the people from Barcelona, were able to pull in information from the French in their language, pull in information from the English in their language, from the rest of Spain in Castellano in their language. And then among themselves, they could speak Catalan. He had an incredible advantage, and that was a life-changing decision on my part. I've got 4 kids who can speak 4 languages and work and write proposals in. But that lesson really was struck home. In my country, it's so bad. I mean, the United States is terrible, and I'm a product of that too. And the trick is, you start the kids off when they're little chumpies, little kids, and you start giving them another language very early. And you give them an advantage, or super advantage. I see this in all of my colleagues that really are functional in another language. They have an advantage over their monolingual colleagues. It's incredible. Anyway, that's what I learned. One of the things I learned there. And I also learned that bottom science is better because I saw that all my colleagues at Ciencias del Mar were generating their own projects on their own ideas. It was curiosity-driven, and they were clearly better in producing more interesting science than my colleagues in Marseille. I felt really bad about that because I love my friends in Marseille. They were good people, but they were getting their directions from Paris, and that was not good. At EOMAR, okay, I mean here one of the things I've learned is exploitation is folly. Just management is the key. Good students, good colleagues, vision, a stable environment are critical to being able to do research. Okay, what have I learned along the way? Teamwork, I've spoken of that. Humboldt's idea is key. Writing should be done daily. Playing with the data spawns idea. All of those things that I talked about, those new concepts that we generated in the EOMAR group, that came from playing with the data. Spending time on Excel, plotting the data this way, that way, making, I don't know, funny looking plots, looking at one part of the data and another part. This kind of thing leads to new ideas and new concepts. The other thing is the need for more scientists. We don't need less scientists. There are people in Madrid that were saying, and this is a couple of years back, oh, we need fewer professors, we need fewer scientists. No, we need more. But we can't afford them. So what we need is more people like Thoreau, more people who are going to be citizen scientists. Maybe their day job is in a bank, but they can have an interest in science and doing it. And this is one of the things that throughout society we're going to have to do this. Why? Because society can only support a certain, an increase, sort of a linear increase in the scientific population. But the knowledge that those scientists are generating is exponential. And what does that mean? It means in the future, each scientist is going to have to know twice, 3 times, 4 times as much as we know now. Their brains? Are they going to be smarter 3 or 4 times? Doubt it. So we've got a big problem. How to solve that? And my feeling is that the way is more citizen scientists. But this problem of knowledge increasing exponentially and the population of science is increasing linearly leads to a real peer review problem. When an editor gets a new, interesting paper, unique, many times he or she cannot find the proper reviewers. Then the science gets reviewed badly. And sometimes papers get deposited in the literature that should not be there. Okay, gatekeep. This is a little thing. This is something I've learned along the way. Be really careful of the gatekeepers and the power brokers. They can ruin your career. Okay? They can just stop you and you have no defense, none. So that's something I've learned the hard way along the way, but it's really something to worry about. Okay, this is Humboldt's, I try to go through fast. Wilhelm von Humboldt, 200 years ago, laid the foundations and set up the University of Berlin, in Germany. His idea was that, it was on a totally unique idea, was that research and teaching go together. Writing, poetry, and literature, and doing artwork goes together with teaching art, literature, and poetry. The professors should be people that practice what they teach. Okay? This is Humboldt's idea. Now, if you look at the top 100 universities in the world, you'll find that the philosophy behind each of those universities follows Humboldt's idea. Personally, I found out, and these are 3 examples, that he's right. Not every time, but many times when I explain something to somebody, when I give a seminar, when I present a poster, I get an idea that I can use in my research. The ETS idea of using ETS came in my master's presentation. I'm talking to the people, and suddenly it hit me in my head, this enzyme I'm presenting that I've spent 3 years with in the succinate dehydrogenase isn't the key. The key is the electron transport system. But the idea came to me while I was talking to the people. The idea that all respiration is based on the ETS came when I was doing a seminar preparation. Right? Again, teaching, the importance of teaching in developing new ideas. The nutrient limitation and enzyme kinetics, the idea behind the enzyme kinetic model for respiration came when I was talking to, I forgot her name, a professor in California. I was at a meeting making a poster presentation and talking to this woman, and I was trying to explain the data, and suddenly I realized that when the pyruvate goes down, so is the substrate, so the electron transport system goes down. That's shutting down the respiration, and that's why the respiration drops, because there's no food. But that idea came to me while I was presenting the work at a poster. The bottom class. Research spawns new knowledge. And then, okay, the research that we do, the new concept is spawning new knowledge, which then goes into the textbooks, which then goes first to the papers, then to the textbook. They go together. Research spawns knowledge, and teaching spawns new concepts. Those things, this is all Humboldt's basic idea, and I believe he was right. What's needed for good science? Curiosity, hunger to know, vision, strong background in basic science. You need to have a foundation. Okay, now I say a protected, energetic team with ambition, love of data processing, skills in writing, speaking, analysis, management, marketing. I say protected up here. And that means you need a leader of the group who serves as the chevalier, the knight, I'm trying to think of the losses. Okay, but you need somebody with a bouclier and an espada, somebody who's going to fight for the group, who's going to defend the group, who's going to try to get money for the group. And that person plays an incredibly important role. Without that person, you can have all the creativity, all the energy and interest in the science, but without the knight protecting the group, nothing. Okay, but all through these things, you see, you need people that they need to be able to write, they need to be able to speak, they need to be able to analyze the data well. Management is key also. Marketing, because even in science, you have to sell your ideas. Okay, it's a marketing thing. Okay, initiative, drive, determination, and discipline. Those are just human qualities that are really, really valuable in being able to succeed. And you need knowledge. So you got to read the literature. Basic science versus applied science. The bottom line is it's all science. Okay. But neither Newton nor Leibniz, they were mathematicians, but I'm lumping mathematics into science here. They had no idea what was going to happen with their ideas on calculus, integral and differential calculus. And yet, in all of the modern technology of today, those two, that foundation in mathematics is critical, absolutely critical. But it started off with these guys just playing around with mathematics. Kepler and Galileo, in their studies of the planetary systems, had no idea of the implications of the atom. And that's the foundation of the whole space exploration program and the military. You know, I hate to say it, but all of the ballistic missiles, all of the artillery, you know, any in modern warfare, you can't. The laws that Kepler laid down and things about Galileo all had implications towards the military and to the space program. In reality, what was basic science at one time, think of Pasteur's work, ends up by being applied science. They're all science. Okay? That's it. Thanks for listening. That's Mount Rainier in Seattle. It's a beautiful place. It's a beautiful place. Dudas, if you have doubts. Okay. Sí tienes preguntas en español. Yo puedo responder en español. This is an example of learning a language when you're older. You know, when you learn a language, you're like, get into by me. But if you learn it as a chicken, and my grandchildren are all learning Spanish and Catalan, and some of them are learning German. But you're learning that. That's the key. Okay.
I only want to say thank you very much. No, really, because I think it's very important that a student can hear about the science like only one thing, not divided into different things, different parts. So, not only oceanography, just science. So, I think your experience is very, very interesting for them, only for me, only for us, but we can speak many times. and many ideas that you have exposed there are really, really important. Ideas about honesty, ideas about certain idealism, I don't know the name.
Idealism, yeah.
So, you know, you have problems with Spanish and I have some problems with English, so.
He comenzado a aprender los idiomas cuando tenía 30, 35 años. Y sí, puedo aprender, pero para mí, en el mismo momento, he intentado de aprender español, francés y catalán. Y yo puedo hablar los tres. Pero con problemas, muchos problemas. Y no puedo escribir un proyecto como los profesores aquí. Y no tengo la competencia en español o francés. En Quebec he hecho mi responsabilidades administrativas en francés. I did it, but mucho, copiar y pegar. Y modificar. But I did it. But la cosa mejor es aprender con los chicos. Y recuerdo en Quebec, era un ministerio de idiomas, me parece. Era un ministerio en Quebec que dice que, es peligroso si nuestros niños aprender inglés cuando están chicos. ¿Qué es eso? But that was a philosophy. That was a philosophy that, we don't want to teach the children until they are 9, 10 or older. And that's bad. The kids, the maximum to learn a language is between 2 and 4. There was a paper in Science which showed that really clearly. And I know myself, because all my kids, not all, but most of my kids speak 4 languages, and they learned them when they were little. They learned them. At the same time, I was trying to learn it, but they learned the language like that, and I learned it like that. So, I mean, it's a real demonstration in my life that when they learn the language, and they don't get mixed up. My kids in the rails in Barcelona, they've got sort of a palanca in their head. They can switch from English to French to Catalan to Spanish, and they do. My daughter, I looked at a video yesterday. She's taking a test to be a professor at an institute, and she gave her presentation in Castellano. But in the home, she speaks Catalan, English, but she did it in Castellano there. And my kids in Germany, I have grandchildren, they're learning German now. Yeah.
Ya que sacas el tema de Cataluña, del catalán, del castellano, hay un grave error en el resto del país, ¿no? Porque en época se consideraba, en principio se prohibía hablar el catalán, ¿no? Durante la dictadura, ¿no? Luego, entonces se imponía el castellano. Y eso iba en detrimento de la formación psicológica, psicoemocional de la gente. Pues yo estuve ahí unos años, conviví perfectamente con gente catalana, casado con castellano.
Sí, muchos.
Y había un entendimiento fantástico. Es decir, no había problema ninguno de comunicación. No se insultaba a nadie, no se le impedía estudiar a nadie por no saber una lengua o la otra. Y eso yo me cuento, no lo que he dicho.
Sí, sí.
Yo que llegué tarde, in Spanish.
Sí, sí.
Yo llegué tarde y creo que teníamos que haber presentado al profesor Ted Packard porque pensé que lo iba a presentar Ciencia Compartida. No sé si todos conocen al profesor Ted Packard, pero creo que lo están estudiando, lo estudiarán posiblemente en oceanografía biológica. Cuando ya han visto que es no solo un experto, es un referente en oceanografía.
Yo había leído cosas sobre él.
Entonces, pues agradecerte esta charla. Yo me emociono muy rápido. Bueno, eso. Agradecerte que estés aquí y bueno, en fin, no puedo decir nada más porque ya ven que estoy un poco emocionada.
Hay una directora, una decana maravillosa. Hay momentos cuando puede tener decanos sin corazón, sin conocimiento, pero Miriam
pero nada, agradecerte que estés aquí.
Estas cosas son importantes porque es curioso. When you go through your competiciones para los puestos, no hay una pregunta o un, how do you say a requirement, requisito, que necesito ser una persona en universidades que quieren enseñar, que quieren dar conocimiento a los estudiantes. Y algunas veces hay personas que ganan puestos que no tienen esa idea, no quieren dar. Ellos quieren tomar. Es malísimo en universidades, porque en institutos, in the whole profession of teaching, the teachers need a corazón que quiere dar.
De todas formas, creo que tú eres una de esas personas que no está...
No sabía al principio, pero aquí en la Universidad de Canaria, no las vamos a Canaria, pero aquí encuentro que me gusta trabajar con los estudiantes porque tengo un placer de dar.
Pues...
Es una cosa,
era
pero no sabía esta cosa al principio, no sabía.
Era lo que les quería hacer llegar a los estudiantes, que no sé si han venido por él o han venido simplemente porque es la charla final, la charla que cierra el ciclo de la ciencia que me inspira. Hemos tenido más o menos éxito, pero creo que el broche final ha sido espectacular. Y creo que a partir de ahora lo van a ver a Ted, lo van a ver de otra manera, quiero decir, porque si realmente lo buscan, verán que es pues, el referente de la oceanografía a nivel mundial. Y lo tenemos aquí, que es lo importante, lo tenemos aquí, no se ha ido a ningún otro sitio, ha venido aquí y se ha quedado con nosotros. Gracias, Ted.
Gracias.